Okay, so today I'm starting the last unit of the year, and this is on inheritance patterns and human genetics. The first thing we're going to look at is a little bit of an extension of chapter 9 when we looked at Mendel and different types of inheritance patterns. There's a scientist named Francis Collins that discovered the gene that's responsible for cystic fibrosis, also abbreviated CF. Cystic fibrosis is often a fatal genetic disorder. What happens is a sticky, thick mucus builds up and blocks the ducts in the pancreas and in the intestines and causes difficulty in breathing. The pancreas is an extremely important digestive organ. In this chapter, you will learn how diseases such as CF and characteristics such as eye color are inherited and expressed.
Hi, and welcome to another episode of Demystifying Medicine. On today's episode, we will cover cystic fibrosis. We will be discussing the basic biological principles of cystic fibrosis, the organs affected, and the various treatment options for this disease. So what do you, a panda, or a neighbor's pet have in common? You're all made up of cells. The cells can be thought of as the subunit of life. Inside these cells are a set of instructions called DNA. DNA is made up of the bases adenine, guanine, cytosine, and thymine. You can think of these bases as instructions that code for genes in your body. For example, the DNA contains instructions for traits such as hair type, eye color, and height. When a nucleotide base is added, removed, or substituted, it is known as mutation. These mutations can change the DNA code, which may affect the traits that are expressed in your body. Now that we know the fundamental genetic makeup of our body, let's move to the next big question. What is cystic fibrosis? Cystic fibrosis is a disorder caused by mutation in the cystic fibrosis transmembrane conductance regulatory gene, known as CFTR for short. This mutation occurs on chromosome 7. This disease is an autosomal recessive disorder, which means that the disease is inherited when a child inherits a defective copy of the gene from both parents who are carriers of the gene. A carrier is an individual who has a defective copy and a normal copy of a gene, and is unaffected themselves. The CFTR gene codes for the CFTR protein, which is an important chloride channel. The purpose of this channel is to pump ions in and out of the cell. The symptoms and associated complications of cystic fibrosis are caused by the dysfunctional CFTR protein channel, which is found throughout the gastrointestinal tract. Affected individuals have elevated sweat electrolyte levels, pancreatic insufficiencies, and lung infections. These signs are observed in most patients, but not all, since severity of the disease can range widely amongst individuals. In Canada, 1 in 3,600 are born with this disease. Studies have shown that cystic fibrosis is more frequent in European Caucasian populations than in any other population around the world. In addition, in the mid-1900s, the median survival age of individuals with this disease was 8 years, but today the median survival age has drastically improved to 30 years, mainly due to better understanding of the disease and improved clinical approaches. Now that we know more about the disease itself, let's talk about what organs are affected and the various treatment options available. Recent studies have shown a higher understanding of the underlying causes of cystic fibrosis at a genetic level. More than 1,500 genetic mutations have been linked to the onset of this disease. However, the most relevant mutation, found in 66% of affected patients, is the deletion of the phenylalanine in position 508 on the CFTR gene. This inherited disease mainly affects the lungs, pancreas, liver, intestine, and sinuses. The most prominent organ affected are the lungs. Excess mucus causes infections and eventual permanent lung damage due to trapped bacteria. Normal digestive functions are also altered by this disease, as the mucus blocks digestive enzymes from reaching the intestines. Therefore, food, specifically fats and proteins, are not digested properly in patients. Finally, inflammation and infection of the sinuses is a common effect of this disease, as this disease affects the layer of epithelium found in the upper respiratory tract. With this advanced knowledge of the causes of cystic fibrosis, a number of treatments have been proposed, with the intent of these treatments being to prevent and control infections in lungs by removing mucus and to prevent blockages in the intestines. A number of treatments for this disease are available, including antibiotics for infections of the airways, chest physical therapy, and other specific medications. Antibiotics are generally the primary treatment for people with cystic fibrosis, as they most likely will have a lung infection. Based on the type of bacteria and the severity of the patient's condition, there are different types of antibiotics that are available, some of which include oral, inhaled, or intravenous antibiotics. A second type of treatment is chest physical therapy, which serves as a way to loosen and remove mucus from the lungs. This should be done 3 to 4 times a day, and it involves repeatedly pounding one's chest and back in order to dislodge the mucus. As it might be physically demanding for some people, there are devices such as mechanical percussors or a positive expiratory pump mask, which both serve the purpose of clearing mucus from the airways. Cystic fibrosis can also be treated by medications, most notably a group called bronchodilators, which help to open the airways by relaxing the surrounding muscles. Finally, as mentioned before, the pancreas and intestines are unable to function efficiently because of this disease. Digestion of food becomes difficult, and eventually malnutrition becomes a cause of the disease. Prevention of malnutrition has become a greater focal point in recent years, as it could help the patient fight a persisting lung infection. Oral pancreatic enzymes are provided to help digest fats and proteins, and supplements of vitamin A, D, E, and K are provided to replenish the fat-soluble vitamins that the intestine can no longer absorb. By implementing these techniques, we can hope to continue lessening the incidence of this disease. That's all for today. Thanks for joining us.
Okay. So, a man named Jeff Pennard worked in Collins's lab, and he actually had CF himself. Collins's group was able to study the CF gene partly because of the work done by geneticists in the early 1900s. Some of this work included experiments by Thomas Hunt Morgan with our friend Drosophila. These are the fruit flies, Drosophila melangaster. He observed that these flies have 4 pairs of chromosomes. 3 of those pairs were identical between males and females, but 1 pair was different in size and shape. In females, the 4th pair had 2 identical chromosomes, which they called X, and in males, they had an X and a shorter one, the Y. Today, these are called the sex chromosomes. The sex chromosomes contain genes that determine the sex. I'm going to delete that now because we understand gender a bit more now. Okay. So the sex chromosome contain genes that determine the sex of the individual. The remaining chromosomes, we know them as autosomes. Humans have the same sex determining chromosomes, XX for female and XY for male. Some organisms like chickens and moths, the males have 2 identical sex chromosomes and the females have 2 different ones. In most plants and in some species of fish, they don't even have sex chromosomes. So a lot of times when, as a society, we're talking about the idea of sex and gender, this is why as a social concept, we talk about gender separately from sex, because some people like to use biology and say, well, females are XX and males are XY, but that's just not the case in all species. So it's important to make that distinction. Like other homologous chromosomes, the sex chromosomes pair during meiosis. As meiosis proceeds, the paired chromosomes separate and move to different cells. This means that the sperm cells have an equal chance of receiving either an X or a Y, while for egg formation, because females are XX, all of the eggs are going to get an X. So what that means is during meiosis, if you start with one cell which has an X and a Y chromosome, at the end of that, you're going to have 1 sperm that has X and 1 sperm that has Y. Whereas for females, you start with XX, so 1 egg will have 1 X each. So then during fertilization, depending on if an X sperm or a Y sperm fertilizes an egg, you will either have a boy or a girl. In mammals, when the egg that carries the X is fertilized by a sperm carrying Y, this results in a male. And if the egg is fertilized by the X sperm, as I just said, it results in a female. In a male mammal, the Y chromosome contains a gene called the SRY. That's called the sex determining region Y. This codes for a protein that causes the gonads of the embryo, gonads being the reproductive organs, to develop as testes instead of ovaries. Because the X chromosome doesn't have the gene SRY, the gonads become ovaries. So what that means is that for the human species and for all mammals, basically, because this is the same throughout all mammals, the default sex of the offspring is female. So that if the Y chromosome is not present, then the SRY gene isn't activated. And so the reproductive organs, the gonads, stay as ovaries. However, if the Y chromosome is present, then during development, the SRY gene will become activated and it causes the gonads to develop into testes. This is the reason why and all mammals, even the males have nipples, even though you don't breastfeed.
My wife is pregnant right now with our first child, and when people see her with her big baby bump, the first question people ask, almost without fail, is it a boy or is it a girl? Now, there's some assumptions behind that question that we take for granted because of our familiarity with our own human biology. For human babies, we take it for granted that there's a 50-50 chance of either answer, boy or girl. But why is it that way? Well, the answer depends on the sex determination system that has evolved for our species. You see, for most mammals, the sex of a baby is determined genetically with the XY chromosome system. Mammals have a pair of sex chromosomes, one passed down from mom and one from dad. A pair of X's gives us a girl, and an X and a Y together gives us a boy. Since females only have X's to pass on in their egg cells, and males can give either an X or a Y in their sperm cells, the sex is determined by the father, and the chance of producing a male or a female is 50-50. This system has worked well for mammals, but throughout the...
Just want to point out, since he said that the sex of the offspring is determined by the males, that historically, as a species, especially when it comes to royal families where producing a male heir is extremely important, females have historically been blamed if the queen doesn't give birth to male heirs. They figured back then, because we didn't know about genetics back then, they thought because the woman was the one that carries the child, that somehow she's responsible for magically determining the sex of the baby. So there have been stories, especially with King Henry VIII of England, who I think he married 8 times. And at least one of those times he was upset that he was not having male heirs produced. And he was very famous for cutting off his wife's heads when he was tired of them.
Of life, we can see other systems that have worked just as well for other animals. There are other groups of animals that also have genetic sex determination, but their systems can be pretty different from ours. Birds and some reptiles have their sex genetically determined, but instead of the sex being determined by dad, their sex is determined by mom. In those groups, a pair of Z sex chromosomes produces a male. So these males only have Z's to give. However, in these animals, one Z and one W chromosome together as a pair produces a female. In this system, the chance of a male or a female is still 50-50. It just depends on whether mom puts a Z or a W into her egg. Certain groups have taken genetic sex determination in completely other directions. Ants, for example, have one of the most interesting systems for determining sex, and because of it, if you are a male ant, you do not have a father. In an ant colony, there are dramatic divisions of labor. There are soldiers that defend the colony, there are workers that collect food, clean the nest, and care for the young, and there's a queen and a small group of male reproductives. Now, the queen will mate and then store sperm from the males, and this is where the system gets really interesting. If the queen uses the stored sperm to fertilize an egg, then that egg will grow up to become female. However, if she lays an egg without fertilizing it, then that egg will still grow up to be an ant, but it will always be a male. So you see, it's impossible for male ants to have fathers. And male ants live their life like this with only one copy of every gene, much like a walking sex cell. This system is called the haplodiploid system, and we see it not only in ants, but also in other highly social insects like bees and wasps. Since our own sex is determined by genes, and we do know of these other animals that have their sex determined by genes, it's easy to assume that for all animals, the sex of their babies still must be determined by genetics. However, for some animals, the question of whether it will be a boy or a girl has nothing to do with genes at all, and it can depend on something like the weather. These are animals like alligators and most turtles. In these animals, the sex of an embryo in a developing egg is determined by the temperature. In these species, the sex of the baby is not yet determined when the egg is laid, and it remains undetermined until sometime in the middle of the overall development period when a critical time is reached. And during this time, the sex is completely determined by temperature in the nest. In painted turtles, for example, warm temperatures above the critical temperature will produce females within the eggs, and cool temperatures will produce a male. I'm not really sure who came up with this mnemonic, but you can remember that when it comes to painted turtles, they are all hot chicks and cool dudes. For some tropical fish, the question of will it be a boy or will it be a girl isn't settled until even later in life. You see, clownfish all start out their lives as males. However, as they mature, they become female. They also spend their lives in small groups with a strict dominance hierarchy where only the most dominant male and female reproduce. And amazingly, if the dominant female in the group dies, the largest and most dominant male will then quickly become female and take her place, and all the other males will move up one rank in the hierarchy. In another very different ocean animal, the green spoonworm, the sex of the babies is determined by a completely different aspect of the environment. For this species, it is simply a matter of where a larva happens to randomly fall on the sea floor. If a larva lands on the open sea floor, then it will become a female. But if it lands on top of a female, then it will become a male. So for some species, the question of boy or girl is answered by genetics. For others, it's answered by the environment. And for others still, they don't even bother with the question at all. Take whiptail lizards, for example. For those desert lizards, the answer is easy. It's a girl. It's always a girl. They are a nearly all-female species, and although they still lay eggs, these eggs hatch out female clones of themselves. So will it be a girl or will it be a boy? Throughout the entire animal kingdom, it does really all depend on the system of sex determination. For humans, that system is a genetic XY system. And for me and my wife, we found out it's going to be a baby boy.
Okay. So during Thomas Hunt Morgan's experiments with Drosophila, one scientist noticed that a single male fruit fly had white eyes instead of the normal red eyes. So red eyes is the normal version of the gene. So naturally what they did was they crossed the white-eyed male with a red-eyed female, which you can see here on the right-hand side with our handy little Punnett square. Remember these guys? Punnett squares. So we have the red-eyed female, here is XX, and the white-eyed male, XY. It turns out when they mated these two together, the entire F1 generation was all red eyes. That determines that red eyes are dominant. That's the key right there. So how did he prove sex-linked inheritance? Well, so the first generation are all, oh, sorry, it was from the previous slide, but first generation are all red eyes. So naturally, then you're going to take, just like Mendel did, you're going to take a female from the F1 generation and mate it with a male from the F1 generation. And you can see what happened is that you got 3-to-1 red to white, but this is the key. White eyes only appeared on males. You never had in the F2 generation, even though you still got the very typical 3-to-1 ratio for recessive trait, but you only find white eyes on the males. So Morgan hypothesized that the gene for eye color carried on the X chromosome, and the Y chromosome doesn't have an allele for eye color. So you've got X with a capital R is red and X with a lowercase r is white. So even if you have a female like this would be considered a carrier because the female has the trait for white eyes, but because it's recessive, her eyes appear red. But if you have a male that gets this, then because there's no alternative form on the Y chromosome, because it's so small, then he's going to have white eyes. So if you cross a homozygous dominant red-eyed female with a white-eyed male, all of the F1 females are going to be carriers, and all of the F1 males are going to have the dominant version of this gene. So let's just do that Punnett square, because we know how to do that. So we've got that, and then we've got XR, XR, and here we've got X little r and Y. So I might have made my box a little bit bigger. Oh well. So this female is a carrier. This female is a carrier, but all of the males you see here, all the males get the red-eyed X chromosome. But for F2, make my box a little bit bigger this time. For F2, you take one of these guys and cross it with one of those. So we've got X big R Y, and then X big R X small r. Now you see we have a difference because here we've got a homozygous dominant female. We have a female carrier here. We have a red-eyed male, but look at this. There's where your white eye comes in because there's no alternate form. So in the F2 generation, half of the females are going to be homozygous dominant red-eyed, half of them will be white-eyed carriers, all of them have red eyes. In the F2 males, of course, you just saw based on the Punnett square, half of them will be red-eyed and half of them will be white-eyed. These results show that genes reside on chromosomes and the red-eyed color gene is on the X chromosome. So these are called X-linked genes. You can go back to that website that I gave you that has all of the chromosome mapping, and you go to the X chromosome. Those are all X-linked genes. Genes like the SRY gene that's located only on the Y chromosome, these are called Y-linked genes. So any trait that is controlled by genes that are located on the sex chromosome are called sex-linked traits.
When eminent Drosophila geneticist T.H. Morgan found an unusual white-eyed mutant fly, he bred pure stocks and tested them. Was the white-eyed character a typical autosomal recessive trait? In a test cross, Morgan...
So I think he's going to bring it up now, but if it was an autosomal trait, meaning that it's not on the X or Y chromosome, you would expect to find an equal number of white-eyed males and females. But because it's an X-linked trait, it only occurs on males.
Pure breeding white-eyed females and pure breeding red-eyed males. If female eggs carried the recessive white eye allele on an autosome and male sperm carried a dominant red eye allele, the mating should have produced all heterozygous red-eyed progeny, males and females alike. Did that happen? In fact, all of the females were red-eyed, but all of the males were white-eyed. The mutant phenotype was apparently linked to the sex of the fly. Morgan hypothesized that this eye color locus was indeed recessive, but was linked to the X chromosome. Female progeny got a mutant X from their mother and a wild-type X from their father, making them red-eyed. Male progeny, however, got a mutant X from their mother and a Y without the eye color locus from their father, and the recessive white eye trait appeared.
So to make this easier, because he's using all kinds of different variables now. So let's just stick with the, if the white-eyed females of that's going to be X little r, and you've got red-eyed male, that's going to be X big R, Y. What happens when you cross those? So you've got X little r, X big R, Y. So what are you going to see? This is why Mendel's work is so important, because you can justify your results by doing the Punnett square. So check that out. All of the females are red-eyed, even though you started with a white-eyed female. And then when you go to the males, look at this. All the males are going to be white-eyed. So you start off with a white-eyed female and a red-eyed male, and then the entire second generation is completely the opposite because it's an X-linked trait. I think that's wild. That's so cool. That's so cool. So the X chromosome, as I've showed you before, is much larger than the Y, so it's going to contain more genes. Most of the X-linked traits do not have a homologue on the Y chromosome. The Y chromosome is just not big enough to hold the same number of genes as the X. And because males have only one X chromosome, a male that has a recessive X-linked trait will exhibit that trait. It will show the phenotype for that recessive trait. Morgan and others hypothesize that if genes are inherited together, it's because they're on the same chromosome. I think there was a question in chapter 9 from one of the worksheets I gave you, which was, what do you think what do you think Mendel would have hypothesized if his genes were closer together? He probably would not have come up with the law of independent assortment and the law of random separation, because genes that are closer together are more likely to be inherited together, because you have, and we're putting together a whole lot of topics that I've taught you over the course of this year. During chapter 8, when we talked about mitosis and meiosis, especially during meiosis, you have the process of crossing over during the synapse, where the two homologous chromosomes line up next to each other and can exchange genetic information. So if that occurs, if you've got chromosomes that are very far apart, they can be inherited independent from each other, but if they're close together, they might be inherited together. So for Morgan, he studied 2 fly genes, one of them for body color and one of them for wing length. They're both located on the same autosome. So you've got gray body, which is dominant to black body, and long wings, which are dominant to short wings. He crossed homozygous dominant gray long-winged flies with homozygous recessive black short-winged flies. The entire generation is going to be heterozygous, right? That makes sense because we've done dihybrid crosses before as well. Now, if we perform a dihybrid Mendelian cross with the heterozygous genotypes, bleh. I hope I have time for this. I think I do. So dihybrid cross, we have to have 1, 2, 3, 4, 1, 2, 3, 4. Now what we put in the boxes, let's extend that. What we put in the boxes, remember we have to pair the first allele from the first gene with the first allele from the second one, and they're both going to be the same. So I've got capitals here. That's our first one. Then we do our first allele with the first set with the second allele. So that's G small l, G small l. Then we do the same with the first, with the second, G big L, G big L, and then G little l, little g little l. All right, so what have we got? We've got all big, and they want phenotypic ratios. So we need gray long, gray short, black long, black short. Okay, so then we've got G, big L, little l, big G, little g, big L, big L. I'm just go through this. G, L, L, G, G, L, L. Little, L, L, big, little, little, big, little, big, little, big, little, big, little, big, little, big, little, and little. La, la, la. All right, so gray and long. So I'm looking for dominant G, dominant L. So I've got 1, 2, 3, 4, 5, 6, 7, 8, 9. I think this is 9331. I think I remember telling you that when you've got heterozygous dihybrid crosses and it's looking for phenotypic ratio, it should be 9331. All right, so we need gray body but short wings. So we need big G's and all little L's. That's 1, big G, all little L's, 2, 3. Black and long, that's little G's but big L. That's 1, 2, 3. Yep, 9331. So that is what we would expect, a ratio of 9331. Then what he did was he crossed 2 of the F1 flies. The flies in F2 were 3 gray long wing to 1 black short wing. So even though we just did a dihybrid cross and we got 9331, when he took 2 of these guys, he got back to our original monohybrid cross ratio of 3 to 1. This is gray is dominant, long wing is dominant. Sorry, I should use the same. This is dominant and long wing, and this is recessive. If the alleles were on different chromosomes, they would sort independently and produce the 9331 ratio like Mendel's P's. Morgan called the pairs of genes that tend to be inherited together linked genes. So we basically what this means is that we expect a ratio of 9 to 3 to 3 to 1, right? We've got gray body, long wing, gray body, short wing, black body, long wing, black body, short wing. We should expect to see all 4 phenotypes in this ratio, but instead of that, we got a 3 to 1, both traits dominant 3 to 1 trait recessive. And that is typical of the Mendelian alleles. So he basically said that the gray body gene is linked to the wing length gene, and a set of linked genes are called a linkage group.
Thomas Hunt Morgan was a geneticist that studied fruit flies. There were 2 traits that he was looking at. One gene determined the body color, and another controlled the wing length. Just like Mendel, Morgan crossed one true breeding line, which was dominant for both traits, with a line that was true breeding for both recessive traits. All of the offspring from this cross showed the two dominant traits, as expected. Then, those offspring were crossed to individuals who were true breeding for the two recessive traits. We would expect that the gametes produced by the heterozygous parent would be found in 4 combinations, and that those 4 combinations would be found in roughly equal numbers. Since the parent that was recessive for both traits could only produce one type of gamete, we would expect 4 different options for the offspring.
So that's what I was talking about. You've got gray body long wing and gray body short wing. You've got black body short wing and black body long wing.
And we would expect them to be roughly equal in number. While
That is
Morgan
not
did
equal.
see the 4 phenotypic combinations, their numbers were not equal. He saw far more offspring that had both dominant traits or both recessive traits than he did having one dominant and one recessive trait.
So the important part about this is that you are seeing that of all 4 different combinations, this is a different experiment than what I just did on the dihybrid Punnett square, by the way. This is more like a test cross because he's crossing it with a homozygous recessive. But this is a second experiment which then further proves the linked gene idea, because you've got here, you got both traits are dominant, and here both traits are recessive. These are far more common than having a dominant version of one of the genes and a recessive version of the other. Same thing here. This one's recessive, this one's dominant. Much, much lower numbers. Anyway, I'm almost out of time, so I want to make sure I finish. So he hypothesized that genes are linked because they're found on the same chromosome. His F2 crosses produced a few offspring, unlike either parent. That's when you saw the gray with short wings. This is having a dominant version of one gene and a recessive of the other, or recessive and dominant. So there were a few, like you saw in the picture that I just showed you, a few recombinations, but most of them were following either both traits were dominant or both traits were recessive. So based on that, this is an important point, because mutations are quite rare. It wasn't enough to explain the unexpected results, because remember, we had 900-ish for all dominant. That's a dumb. And 900-ish of all recessive. And then there were like 200-ish and like one something for the weird combination. So they do occur, but far, far less than having both traits dominant or both traits recessive. So he inferred that crossing over was responsible. During the first division of meiosis, crossing over does not create new genes or delete old ones. It just rearranges allele combinations. All right, last thing. The farther apart 2 genes are on a chromosome, the more likely crossing over will happen. Again, if you imagine crossing over, if 2 genes are right next to each other on the chromosome, and they're more likely to be inherited together. The farther apart they are, the more likely it is that you can have crossing over and you can have new versions of those genes appear in future generations. The greater percentage of F2 showing recombinant traits, the farther apart the genes for those traits must lie on the chromosome. So the more variety you see, the farther apart those genes are. That's what that means. Researchers do breeding experiments and use the results to prepare what's called a chromosome map. This is a diagram that shows the linear order of genes on a chromosome, like so. That website, the link that I gave you for the Human Genome Project, it has all the chromosome maps for human chromosomes. So the percent of crossing over for 2 traits is proportional to the distance between them on the chromosome. So the closer they are, the more likely they're not going to cross over, and the farther apart they are, the more likely they are to cross over. A map unit is a frequency of crossing over of 1%. So 1% chance of crossing over, that is equal to 1 map unit. Okay. I am not getting into mutations today.